Assalamu alaikum everyone. In this video lecture, we will be discussing some tips and tricks of high power drawing of cells. So guys, whenever examiner asks you to draw high power drawing of cells, what you need to do is that you need to observe your specimen at high power. That is, your objective lens should be into 40. So whenever you observe your specimen at high power, you will be observing a lot of cells. For example, this is the specimen in front of you at high power, right? And you can see a lot of cells. So you don't need to draw all the cells that you see. You will need to follow the instructions of the examiner. Examiner will tell you that you need to draw which cells. For example, over here you can see the question that draw three xylem vessel elements which are touching each other. So guys, you have to carefully follow the instructions. For example, this is the specimen of dicot stem, and you can see that this is the xylem tissue. You have to draw three xylem vessel elements which are touching each other. So can you draw these? No, you cannot, because these are three xylem vessel elements, but they are not touching each other. Why? Because cell 1 touches cell 2 touches cell 3. We can say that cell 2 is touching both cell 1 and cell 3. But these three cells are not touching each other. So guys, you have to be very, very careful. You have to follow the instructions. So we need to find the cells, three cells which are touching each other. For example, we can draw these three cells because these three cells are touching each other. So instructions need to be carefully followed while drawing the high power drawing of cells, right? Let's draw these three xylem vessel elements. So guys, when we are drawing these cells, what we need to be careful about? We need to be careful that the lines are clear and continuous, right? And we need to carefully draw the cell walls, and we just need to draw what we see and not a textbook diagram, right? And while we are drawing the plant cells, we need to make sure that we carefully draw all the cell walls and we carefully draw the middle lamella. So whenever you draw the plant cells, don't draw the cell walls initially, just draw the plant cells, and then after that, draw the cell walls once you are done with the plant cells. So the cell wall should be drawn like this. It should be inner layer like this. The outer layer was drawn previously, now you will just draw the inner layer. And when you draw the inner layer, the middle lamella will be drawn automatically. Let me give you an idea. So guys, this is the middle lamella and it's drawn automatically, right? Because the examiner in this question is not asking us to label, so we won't be labeling any diagram. We won't be labeling these cells, right? So this is how we draw the high power drawing of cells. Because these cells are dead, there is no nucleus, so we won't be drawing any inner details of the cell because there are no inner details. But you can be asked to draw living plant cells, and those cells will have inner details like the nucleus and maybe the chloroplast as well. So you need to draw that, and you can ask, you can be asked to label the drawing by the examiner. For example, if we label this diagram, so how can we label this? We can label this as the cell wall. So guys, whenever you label, please make sure that you don't draw arrows, right? Just draw a simple label line, and your label line should be short, right? It should not span the entire diagram. For example, if you want to label the cell wall, you won't label it like this. This is wrong. Draw a short label line which does not span the entire diagram. And because this is a xylem vessel element, this is the xylem vessel element, this does not contain the cell membrane. You can label this as the lumen or space of the xylem vessel, right? Okay. What else you need to be careful about is that there should not be multiple strokes. So over here, you can see that there's a multiple stroke. You need to avoid this. And somewhere, so at some points, there will be broken lines. So avoid broken lines, avoid multiple strokes. I know it's difficult, but you can do it. Just erase and avoid broken line. Over here, you can see that there's a broken line. Avoid this broken line. This is how we draw the high power drawing of cells. So guys, let's just go through quickly through all the rules that we just discussed. Draw exactly what you see, not a textbook diagram. Clear and continuous outlines should be drawn. Avoid faint outlines, broken outlines, and multiple strokes. Only label if you're asked by the examiner. While labeling, draw horizontal label lines using a scale. Do not make arrows. Drawing and label lines should be made using a pencil. Label should be written with a pen. So, guys, this is an important point. So, what you need to do is that while you need to draw your drawing using the pencil. You have to draw the label lines using the pencil as well. But when you put the label, it should be done with the pen. Right. Label lines should be short and not span the entire drawing. This is what I already discussed a while ago. Also, label lines should not intersect with each other. You need to be careful that when you are labeling, so for example, if you are labeling the cell wall and the lumen, so these label lines should not intersect each other. This is wrong. Moreover, drawing should be large. It should cover most of the cell space, sorry, most of the given space, right? It should cover more than 75% of the space. Leave the space on either side for the labels. Over here, you can see that I have drawn the cells and I have covered most of the space given to me. And I kept some space on the left side and on the right side, and some space at the top and some space at the bottom for the labels. You need to be careful about that. While drawing cell walls, draw middle lamella. That's what I discussed, that we need to be careful. We need to draw the middle lamella. That is the layer between two cell walls of the plant cells. Okay, guys, thank you so much for watching. Allah Hafiz, everyone.